How do new discoveries reveal raw immune response secrets? Imagine unlocking a hidden code that explains how your immune system sometimes turns against your own joints. Recent scientific breakthroughs are doing just that, shedding light on the complex immune responses behind rheumatoid arthritis. These discoveries are helping scientists understand what triggers inflammation and how we might better diagnose and treat this condition in the future. At the heart of rheumatoid arthritis lies a breakdown in immune system regulation. Normally, helper T cells act as the commanders of immune defense, coordinating responses to infections. But in rheumatoid arthritis, these cells become dysregulated and start attacking the body's own joint tissues. This misdirected attack causes inflammation, pain, and joint damage. Researchers estimate that around 18 million people worldwide live with this condition, and understanding how this immune misfire occurs is a major focus of ongoing research. A recent breakthrough from Kyoto University identified a molecule called IgFL2, which is produced by a specific type of helper T cell found in the joints of rheumatoid arthritis patients. This molecule appears to regulate inflammation within the synovial tissue, the lining of the joints. Because IgFL2 is specific to primates, studying it helps scientists understand human disease better. It could serve as a marker for disease activity or even become a target for new therapies, helping to control inflammation more precisely. This discovery is a big step forward because it pinpoints a specific molecular player involved in the disease process. Scientists are also learning that rheumatoid arthritis involves a complex web of inflammatory mechanisms, not just one pathway. The environment inside affected joints becomes hypoxic and acidic, which promotes the infiltration and activation of immune cells. This environment helps explain why inflammation becomes chronic and progressive over time. Multiple immune cell types are involved. Macrophages, which are cells that process foreign substances, can trigger further inflammation when exposed to oxidative stress. T cells recognize antigens in the joint and releases cytokines that promote ongoing inflammation and tissue destruction. B cells, although fewer in number, produce autoantibodies like rheumatoid factors and anti-citrullinated protein antibodies. These autoantibodies form immune complexes that deposit in the joint lining, fueling inflammation. Additionally, mast cells in the joint releases inflammatory mediators such as tumor necrosis factor alpha and interleukin-1 beta, which further worsen joint damage. Recent research has also clarified the role of autoantibodies in rheumatoid arthritis. These autoantibodies target altered proteins in the joint, especially those modified through a process called citrullination. During citrullination, enzymes convert arginine residues in proteins into citrulline, changing how the immune system recognizes these proteins. This process creates new targets that the immune system mistakenly sees as foreign, especially in people with certain genetic backgrounds. Autoantibodies like rheumatoid factor and anti-citrullinated protein antibodies then form immune complexes that deposit in the joints, triggering inflammation and damage. Genetic studies have revealed that specific immune responses are linked to the development of rheumatoid arthritis. For example, certain proteins circulating in the blood have been identified as having a causal relationship with the disease. One protein called KCNIP3 appears to have a protective effect, which could lead to new approaches for prevention or treatment. Interestingly, scientists have found signs of immune activity years before joint symptoms appear, opening the door for early diagnosis and intervention. Beyond adaptive immunity, new research emphasizes the importance of innate immune system activation in rheumatoid arthritis. Pattern recognition receptors detect signals from pathogens or damaged tissues, activating immune cells like macrophages, dendritic cells, neutrophils, natural killer cells, and mast cells. These cells release cytokines, activate the complement system, and present antigens, all of which contribute to ongoing inflammation. Modern treatments are now exploring ways to regulate these innate immune responses, aiming to control inflammation more precisely rather than broadly suppressing the immune system. All these discoveries are crucial because they help address the challenge that about 30% of rheumatoid arthritis patients do not respond well to current therapies. By understanding the specific molecular pathways involved, researchers can develop more targeted treatments that focus on the underlying causes of inflammation. Early diagnosis is also becoming more feasible with markers like IGFL2, which could lead to personalized treatment plans based on an individual's immune profile. As research continues to evolve, scientists are working to find ways to calm the overactive innate immune responses while also addressing the autoantibody-driven aspects of the disease. These advances promise a future where treatments are more effective, personalized, and capable of preventing joint damage before it starts. If you or someone you know is managing rheumatoid arthritis, staying informed about these scientific developments and working closely with healthcare professionals can help you make the best choices for your health.